So I would like to welcome everyone to MTRI's June Sit-Back Seminar Series. My name is Morgan Snare, and I am a Summer Research Assistant here at the Mercy Tobiatic Research Institute. But before we begin, I would like to acknowledge that MTRI is in and we are meeting from Aphid-like, one of the 7 districts of Mi'kma'ki, homeland of the Mi'kmaq people. We acknowledge the treaties of peace and friendship, and we thank the Mi'kmaq people for their generosity in sharing their homeland with us. MTRI is a research-based nonprofit nestled in southwest Nova Scotia, near Keggwee National Park and Historic Site, and within the Southwest Nova Scotia Biosphere Reserve. Our mission is to promote, conserve, and sustain biodiversity in Aphid-like and beyond. Tonight, I am very pleased to introduce Dr. Christopher Egg. Chris is a research scientist with Natural Resources Canada, Canadian Forestry Service, and an adjunct professor in the Department of Biology at the University of New Brunswick. Chris's research aims to understand how forest ecosystems respond to disturbance at broad spatial and long temporal scales. A major theme of Chris's research is understanding the direct and indirect environmental effects of pesticides on non-target species. Chris is passionate about the conservation of species at risk and volunteers for several local and national conservation groups. Next, I will hand the seminar over to Chris, but I would like to remind everyone to please keep your mics muted, and if you have any questions during the talk, please type them into the chat window or wait until the question period at the end. And Chris, I'm going to hand it over to you now.
That classic video conference mistake or video presentation mistake when you don't unmute yourself. Good evening, everybody. I'm Chris, as you just said, and I'm speaking to you from Fredericton, New Brunswick, which is located in the unceded and unsurrendered territory, traditional territory of the Wolastoqey people. And I'm very pleased to be here to speak to you tonight about imidacloprid and its persistence in the environment after it's been used for the treatment of Hemlock Woolly Adelgid. And if somebody could just give me a quick thumbs up to let me know that my slide's shared, that'd be fantastic. Thank you very much, Morgan. And so what I'm going to talk about tonight has been some research that's been ongoing down in Nova Scotia at 2 study sites. And it's all around the control of HWA, which is a major pest or invasive pest for hemlock trees. It's a large collaborative project that some of you may have heard about or been a part of in some capacity. So there's a group of us that are working on this from across both provincial, federal governments, several universities, some industry partners, and some other local groups as well. And there's too many people to list on a title slide, so I'll put them as my second slide to say that the project simply is not possible without a large number of people working on it. So hemlock trees, or the eastern hemlock, is a long-lived, slow-growing, shade-tolerant species, and it's a very important part of the ecosystem. And it's a remnant forest because it produces stands of old, closed canopy forests and very damp and humid, and they're very important for many components of the ecosystem, such as, say, cold water stream communities and other shaded insect communities. And so with these old growth forests, there's a very much concern about the loss of these forests and their importance to the regional ecology. And a major threat to east hemlock is the Hemlock Woolly Adelgid. It's an invasive pest species that originated in Japan. There is an adelgid that's native to the west coast of Canada, but this prickly species from Japan. It was first detected in Virginia in the 1950s, and it invaded Nova Scotia within the last 5 to 10 years. The big issue with this is that once a tree is infested with HWA, it's most likely going to die within 14 to 15 years. And there's been a large body of work, lucky for Canada, but unlucky for hemlock forests, a large body of work on the eastern coast of the US, where the species first invaded, on different types of control mechanisms and control methods that might work for controlling the species. And so our work here in Canada has been able to borrow upon some of this work down the east coast of the US, and to look at techniques that are effective and rolled out of a large number of ineffective techniques. For the control and mitigation of any insect pest, you have a large number of controls or techniques that you could use. One of them is chemical control, which is generally a short-term technique that you can use to prevent mortality of trees. And then there's other techniques such as biological, which will provide a long-term strategy for controlling these invasive species, but it's very much at the research stage right now for Hemlock Woolly Adelgid. There's some other possible techniques that may come into work. So there's some silvicultural techniques around thinning trees and adjusting stand densities and that type of thing that may limit the spread. And then there's two other pieces. So genetic resistance, which involves breeding resistance within trees. That's another very long-term strategy. And then there's an ARC strategy or conservation strategy, which would be to conserve genetic diversity of high-value trees within an ex situ environment. In order that if we can control hemlock or Hemlock Woolly Adelgid in the future, then you could reintroduce hemlock trees. But this doesn't solve the current problem of HWA in the landscape. One of the big strategies right now that's been ongoing is chemical control. And really, this is a stopgap method. We know, and well, I say we know, but a lot of the researchers and the people who are working on this problem know that this is not going to be a long-term strategy. And it's for two reasons. One is the cost, and it's not possible to treat every tree. And the other piece would be there's risks that are associated with using chemicals in the environment that are unsuitable in some cases. So chemical control in general is never the first choice for controlling invasive species. It's sometimes the only choice that we have. And in the case of hemlock, it very much is right now the only choice that we have for a short-term stopgap measure. When we're using chemicals for the control of any particular insect or really any, so the same thing we need for herbicides, there's a preference for narrow spectrum specific treatments. And this is both the mode of action where it's only going to affect certain species, the target species. The application method gets that chemical only where it needs to be. And then we look at both space and time, where the effect that the chemical has is within a defined space and a defined time. And so these general principles are those that would then reduce the risk of using chemicals in the environment. But they don't remove all potential risk. So with any sort of thing, especially with the environmental use of pesticides or any action that we take on the earth, there's always risk associated with it. And a good example of this is vehicles. So driving cars and operating vehicles or being around cars poses a risk to harm or death. And so we know this is the case where we have car accidents all the time. We know there's risk associated with them. So we put rules and regulations around how those vehicles are used and how people can interact with vehicles. So there's speed limits, there's stop signs, there's separation of pedestrians on sidewalks or on the side of the road and cars on the road. And that limits the risk. And the case to make would be the logical conclusion of that is that when a person chooses to interact with a vehicle, that they've accepted that risk, or they say that risk is then acceptable to them for the goal that they want to achieve, say going to the store or what have you. So we would say that if used properly, vehicles have a low likelihood of risk, or there's an acceptable risk associated with their use. Now, for environmental toxicology or the use of pesticides, it's made up of two things. So environmental risk is really, in its essence, the toxicity of a particular compound and the exposure that would occur in the environment. Toxicity is made up of two things. So it's made up of the dose and the effect. So as you increase the dose of a compound that something is exposed to, you then expect to see an effect occur. So at low doses, we don't necessarily, at low doses of a potential chemical, we don't expect to see an effect. Eventually, the concentration or the dose that something is exposed to elicits a response, and then everything starts to respond, or more individuals begin to respond until we reach the top of the curve, at which time everything is responding to it. There's two important variables that we can draw off this curve that will come a point later in the talk. The first is a LC50 value. This is the lethal concentration at which 50% of organisms respond. That's just to be derived from a laboratory study. And there's a no effect concentration. And this no effect concentration is the lowest concentration tested at which no effect has been observed. So these two values become important. The LC50 value is a way to rank chemicals for potential toxicants or how toxic something is. And then that no effect concentration is the value at which you wouldn't expect anything to respond in the environment at that value. So one is a way to rank chemicals, and the other one is a way to predict when nothing would happen in the environment. These values are derived from a large number of laboratory studies or simulated ecosystem experiments, lab studies, mesocosms, microcosms, whole egg experiments, all kinds of things to derive these toxicity values that are then used for predicting where effects might occur. The second piece of that is exposure. And so when we think about exposure, we think about the scenario in which a chemical is used in the environment, and then the scenario in which that chemical then becomes present in the environment. So for imidacloprid, the technique that is used right now is bark spray. So it's sprayed from a backpack sprayer onto the bark of the tree. So you can see that there's a photo there on the left-hand side of one of the colleagues from CFS, I think, spraying a hemlock tree with imidacloprid. And when that spray happens, the tree then takes up the chemical and it enters into the tree. And then once it's into the tree, the exposure route to the environment could be through falling litter, so needles that are falling out of the tree that contain imidacloprid. And from that, we would then have questions. So how long after application does it take to get into the needles? How frequently is it in the needles? And how much of the chemicals in the needle? And the other way it could enter the environment is through the soil. So it could enter the soil in two ways. So the first would be runoff. So it could, say a rain event happens after application, the chemical washes off the bark and enters the soil, or the tree could transport the chemical in the opposite direction. So from the outside of the bark into the tree and then down into the roots and into the soil. And from here, we would have the same number of questions. So how frequently does it enter the soil? How much of it is it in the soil? How far away from the tree does it move? And how long is it present in the soil? So these are giving us the ideas of how much is present, how long it's present for, and where is it present in the environment after application. The next piece of this is to look at what the chemical is and to get an idea of where or how far away we should look. So imidacloprid is a neonicotinoid insecticide. It's commonly referred to as a neonic, is a common term that you'll see in common literature and news stories and that. It's a non-selective insecticide, so when it comes in contact with an insect, it will kill the majority of insects. It has low soil absorption, so what that means is it doesn't bind strongly with soil, and high water solubility. So it won't bind with soil, and it will dissolve in water relatively easily. It breaks down due to sunlight, so UV light causes imidacloprid to break down into a series of compounds, one of them being olefin, which can be more toxic than imidacloprid itself. And then it persists in the soil. So the time it takes to degrade in the soil, a half-life in the soil would be 16 to 456 days, which is quite long. So it takes a long time to degrade in soil. This is likely because sunlight doesn't penetrate soil. It just sits on the top, or doesn't get into the soil very deep. And in water, that half-life is less than 5 days, likely because sunlight can penetrate water and break down the compound in the water. So what this tells us is that if imidacloprid does enter the soil, it's likely to stay in the soil for a period of time, and it does have the potential to move because it's water soluble. Now, the use of imidacloprid has raised environmental concerns. So the Pest Management Regulatory Agency, which is the branch of Health Canada that determines which pesticides can be used for what purposes. In 2016, they concluded that there was a risk to aquatic invertebrates from agricultural use, and have begun the phase out of using imidacloprid in the agricultural environment and our agricultural application scenarios. Now, this is largely because repeat applications of imidacloprid, so annual applications to the same soil areas, has resulted in continuous runoff into aquatic environments. So once it gets into the water, it breaks down very fast. But if you have a continuous runoff, it has the potential to be there for long enough to have an effect. The PMRA also reevaluated imidacloprid effects on pollinators in 2019. And what they found there was that there is a risk to pollinators when it's used on attractive species. And these are attractive species of plants. And they're referring to species that are attractive to pollinators. So these are flowering plants and things that are pollinated by bees, not wind pollinated. So hemlock trees are largely wind pollinated. So this risk, they're not a big source of food for pollinators. So it's unlikely to have a risk to pollinators in that case. So taking this information, we then constructed some large-scale field trials at McKay Lakes and Sissiboo Falls in Nova Scotia. At each one of these sites, we took 4 1-hectare blocks One of those blocks was treated, and they're paired. So one is treated and one is control. And so there's two maps here showing the study site. The yellow areas, the yellow circles are untreated, and the green circles are treated. And within those treated trees, or those treated blocks, we use Zytec, which is the chemical, the formulation name, the proprietary name of imidacloprid formulation, and applied it to 40 to 60 mature hemlock trees in that block. And that was done twice. So 40 to 60 trees were treated in 2020, and 40 to 60 trees were treated in 2021. Now, within these areas, we took both Adelgid counts from taking branch samples from the tree, and using those same branch samples, we used the foliage and ground up the needles from those branch samples that were collected from the trees to look at how much imidacloprid is in the needles and could potentially be falling to the ground. And then measured soil from the 5 largest trees, both 50 centimeters and 100 centimeters away from the tree, taking soil cores on the downslope side. So these, if you picture a tree, we focus on the downslope side, so where the slope is going away from the tree, to try to measure where that concentration would most likely be highest in the environment to capture a worst case scenario. So the Adelgid counts, what those showed, and this is some work by Dr. John Sweeney with us in Fredericton. What he found is about 5 months after application, there's no effect. So Adelgid counts largely remain the same between the treated trees and the untreated trees. But 12 months or 1 year after treatment, the HWA densities are a lot lower on the treated trees than the untreated trees. And this is exactly what we would expect from imidacloprid, that it has an effect on the adelgids on those insects, and exactly why the chemical would be used. It also matches some of the data coming out of the US that shows that imidacloprid takes approximately 1 to 2 years to actually start to have an effect, for that tree to move it up to the needles, to the buds that the HWA are feeding on, and then for it to have a population level effect on the adelgids in the trees. So this is good news because the treatment is working, or does appear to be working on the adelgids. Now, the residue samples, this is just to give you a map or a quick table of all the data that was collected. So 6 months after application, it was collected at 50 centimeters from the tree and then in foliage. And then 10 months at 50 centimeters and 100 centimeters. That difference there is because of COVID. We weren't able to travel down to Nova Scotia to collect samples, so we had a partner do it. And they were only able to get one set of samples or one distance. And then at 12 months from the tree, 12 months after treatment, collected samples at 50, 100, and then in foliage. Those samples have been analyzed, prepared and analyzed, and I'll show you the data from that. And right now, we're in the process of collecting samples at 18 months. That just happened in May, and we're processing the samples in the lab right now. So I don't have that data available to you. You'll notice that there's an extra rows here for soil at 200 centimeters in the tree and 400 centimeters. And I'll come back to that in a second and tell you why that was done. So first thing, the foliage data. Within the trees, 6 months after application, there was no detection in the needles. And this matches what was found in the US, in that it doesn't show up higher up in the tree. It's not transported in the tree. It takes about 12 months to get up into the crown of the tree where it would have an effect. Surprisingly, at 12 months after application, we only had 2 detections in the 80 samples that were run. This is really low and was pretty unexpected. And these data match another set of data that we've collected from trees that have been treated in Ontario, where it's an equally low detection rate within the trees. And so this begs the question of, well, the adelgid counts were lower, and I showed you that data, but where's the imidacloprid? Like, why aren't we finding it in the needles? And there's a hypothesis that we're thinking right now is that the adelgids themselves don't feed on the needles; they feed on the buds and on the shoot tissues where they feed. So perhaps the imidacloprid is just in the branches or the twigs. It hasn't actually been moved into the needles. And so we're processing some branch samples this year to see if it's present there and see if that explains why we're not finding it within the needles, but we're still seeing this effect on the adelgids. So it remains an interesting open question as to what's happening. Now, it's a very different story in the soil. So these are the soil samples, and this is just the percentage of samples that it was detected in. both time application, the distance from the tree, and then the two compounds. So it's imidacloprid, which is the active ingredient, and then there's olefin, which is that major breakdown pathway, breakdown product. And there's some data in the literature that says that olefin may in fact be more toxic than imidacloprid. So that's why we're measuring it. The big take-home message from this is that basically after treatment, we're detecting imidacloprid in every sample. After application, all the soil samples, so between 60 and 100% of our samples. And olefin, we're seeing a similar trend. So 50 centimeters from the tree, it's in about half the samples, and then 100 centimeters from the tree, it's a little bit less, so 2.2 and 22.5%. And this is probably to do with the temporal piece, where it's taking the imidacloprid has to move, and then the imidacloprid has to break down to be able to detect olefin. So that's likely what's happening. So the take home or the important message here is that imidacloprid and olefin are found around the majority of trees that are treated in the soil. And this is interesting and was somewhat surprising because it was a bark spray that was being applied. This is what the concentrations look like. So this is imidacloprid and this is olefin. These figures are squished because of those outliers. So there's an outlier, very high concentration measured at McKay Lakes and as well at Sissiboo. So I'm just gonna remove those outliers. I'm not going to adjust the data at all, but I'm just going to shift the y-axis so we can get a better look at the concentrations after with distance from tree. And so when I do that with imidacloprid, there's a very clear signal that if you look at both the 50 centimeters from tree and the 100 centimeters from tree, that there's a decline. So it declines 6 months to 10 months after application and then remains relatively stable. There's this weird blip that's going on at Sissiboo where it's down and then it's up. We think that has to do with where the samples were collected. And we see a similar pattern within the olefin concentrations where it declines at McKay Lakes, 6 to 10 months, stays relatively stable. And then we have that weird little blip going up. And then statistically, when we look at this at the Sissiboo site, olefin is staying stable at 50 centimeters from the tree. From 50 centimeters to 100 centimeters from the tree, there's a rapid decline in concentration. So the majority of the imidacloprid is staying close to the tree within 50 centimeters, and there is some moving out to 100 centimeters from the tree. And so this information now gives us an idea of how frequently it's present. So it's present in the majority of soil samples. And now we have an idea of how much is present within the soil samples after application. But the question now shifts over to, should you be concerned or is there any worry about the amount that's found? And so what I conducted was what's a preliminary risk assessment and calculated risk quotients. Now, risk quotients are a really simple way to look at whether or not you would, whether or not there's actually environmental risk. So what this is we take the concentration that's measured in the environment and then we divide it by a critical value. If values are greater than 1, it predicts risk and the need for further study and context to place around it. If values are less than 1, the conclusion would be that we wouldn't actually predict any risk really at all. And so this method of using risk quotients is really a way to screen out compounds where there's unlikely to be any risk, and so there's no need to follow up studies rather than predict actual risk. Now, this is because we use the most sensitive organisms to do these studies. So for acute exposures, we use that lowest LC50 value, so the lowest value tested or determined in the literature. This is that lethal concentration 50 from a lab study. And for the chronic exposure, we use the lowest no observed effect concentration ever measured in the literature. So these values are determined to be conservative by nature in order to say whether or not future studies are needed. So the critical values that were measured were for the 14-day LC50 using an earthworm species. That's a laboratory earthworm species. And that was found to be 0.7 milligrams acid equivalents per kilogram soil. The chronic NOEC concentration, so that no effect concentration, is earthworm reproduction. So that value is lower. And then for pollinators, a nesting bee species was used. There's actually not a lot of data on bee species out there, a nesting bee species or soil nesting bee species that would actually be exposed to imidacloprid. So this value is really low for that reason. So when we calculate those risk quotients, this is the data. Don't bother looking at it too much because I'm going to walk you through it in the next couple slides. So basically, we have an acute exposure, a chronic exposure in the pollinator, and then we're looking at time, distance from tree, and then calculating a risk quotient for the mean value that's detected, and then the upper 95% concentration of concentrations that have been measured. So I'm just going to show these up in blue. So I'm going to highlight the values in the acute exposure category that are greater than 0, and I put those in bold. And so these are the, at 50 centimeters from the tree, the risk quotients would predict that there is risk, 50 centimeters from the tree. The chronic exposure, all the values are bold, indicating that at all distances from tree, there is risk, both using the mean value and that upper 95% confidence interval. And for bees, it's the same story. So for pollinators, that mean value is above 1, and the upper 95% confidence value is also above 1. You can't actually interpret the magnitude of these values to say predict increased risk. It's not the way these values work, because we are using those lowest. We're intentionally meaning this to be very conservative. It really is a categorical piece where values greater than 1 predict that environmental risk could occur, and values less than 1 would predict no risk. So if the values are less than 1, this talk would end now, and I would say there's absolutely no risk of using imidacloprid in the environment. That's not the case, obviously, here. So we need to talk about what these values actually mean, and talk about how we're going to get a little more context to understand what that risk actually means. So risk quotients, the context around them is that they don't predict effects, they predict risk, right? So there's a difference there. They're not saying something will happen, they're saying that there's risk that something could happen. They are conservative, they're using the most sensitive organism, they're assuming continuous exposure and absolutely no mitigation measures present. And what they're really indicating is that there's a need for more experimental field and operational monitoring studies to further place context around what that risk means. So now I'm going to talk about these October 2020 applications. So these were 2 years ago, 18 months after. So these would be 18 months after in May 2022. Because of that risk up to 1 meter from the tree, that's why we increase these distances out to 2 meters and 4 meters from the tree. We really want to know how far away from the tree the chemical is moving, so we can better contextualize that risk for particular application trees. That's why this data becomes really important to know actually how far from the tree is it moving. We know it's 1 meter. Is it 2 meters? Is it 4 meters? We hope to have that data back very soon. Now, of course, there's a gap here, and the piece would be, okay, well, what about that 6 months and 12 months? What about that first year after application? Showed you that imidacloprid does break down in the environment, so the concentration would be lower. That's where the samples that were collected in 2021 come in handy. Or sorry, from the 2021 application. So there was another application made to another set of trees in October of 2021. And so my group's been back and have collected samples at 50, the entire series of distances from the trees after this application period. So we will have that data for 6 months and 12 months after to be able to fill that gap in with these values once they become available. The timeline for this is we're processing the samples now. We'll get them shipped to the lab in probably a month or two. The data will be back sometime in the fall. And so I'm more than happy to come back and give a shorter version of this talk and sort of update a couple slides with these new data when they come in and make them available to folks. So the preliminary conclusions from all of the study would say that yes, bark spray of imidacloprid is likely reducing the number of adelgids on the hemlock tree, so it is achieving that effect, the desired effect. Surprisingly, neither imidacloprid or its breakdown product were detected in foliage 1 year after application. And I talked a little bit about whether or not this is because the imidacloprid has been moved into the needles, or if it's just present within the tissue that the adelgids are actually feeding on. Imidacloprid and olefin were detected in most of the soils up to 1 meter away from trees. And those values that we're detecting do predict that there is possible environmental risk occurring. And what that means, it's not predicting that effects are going to occur. What it's predicting is that we need a little more information to understand how the chemical is behaving in the environment before I can provide a more comprehensive statement of that. And so what that means is we need to understand the spatial distribution, so how far away from the tree it's moving up to that 4 meter distance, and how fast it's breaking down in the environment at different distances from the tree. And as we get more data in, we'll be able to provide these values and hopefully aid folks in making the decision about prevention of the expansion of Hemlock Woolly Adelgid. And so I'll end there, and I'll put my contact information up, my email, and I'll leave this up there. If anybody has any questions that they want to follow up with, feel free to reach out to me through email. If anything comes up after the question period here, or ask me any questions now, it's happy to be here. Thank you very much for listening.
Awesome. Thank you so much, Chris. So if anyone has any questions, they can feel free to put them in the chat or turn their mic on. I see there's one in the chat here right now that I will read out. So this one is from Tom Rogers, and it says, Chris, what about stem injection?
Yep. So stem injection was done on another series of trees. From the environmental fate perspective, because it is injected into the tree, we wouldn't expect it to enter the soil, because it's entered into the tree and then moves up into the foliage. Unfortunately, we don't have environmental fate data from that, so I can't talk too much about what happens there. I would kind of expect the same result, though, about it not entering the needles or not being found in the needles 1 year after application. The trade-off with the stem injection versus the bark spray is that the stem injection treatments are really expensive. They require somebody going out, drilling a hole in the tree, putting a pressurized cartridge in the tree. It takes time, it takes a lot of money to do it. It's a lot of person power to do that, versus the bark spray, which is being able to done from a backpack sprayer. One person can do multiple trees within a short period of time.
Awesome. Sarah asks, is there evidence of imidacloprid in regeneration of species other than eastern hemlock?
Oh, that's a really interesting question. So there is some work by Elizabeth McCarthy in the US who looked at imidacloprid in flowering plants or plants that were growing up around hemlock trees that were treated. Now, these hemlock trees in the US, they use soil drench. So they basically dig a trench and they put a powder in that trench. So they're applying it directly to the soil, is one method that they use. And so she has found imidacloprid within those plants that are growing up around the hemlock trees, but it's at very low concentrations. So it is there, but the concentrations are considerably lower than what we're measuring here.
I have a follow-up to that question. Sorry, I just don't want to.
No, go ahead.
If there was other, let's say it was present in the regeneration of another tree species, an example being, oh, I don't know, spruce. You said that imidacloprid is effective on most invasive insects. So would it help with like spruce budworm, for example?
Oh, I actually don't know the answer to that.
Oh.
I mean, theoretically, yes, because the spruce, if it was in the soil, the spruce trees could pick it up, it would move into the tree, the budworm are feeding on the tree. So theoretically, yes, but I don't know. I don't know if it would be effective.
Right.
Yeah, I don't actually know. Theoretically, yes, but I don't know. And I can't think of anything. Nothing comes to mind for that type of treatment. With budworm, it's probably partially because there's a lot more effective treatments, and there's treatments that are very narrow spectrum. So like BTK and tebufenicide are the two. They're very specialized and very specific insecticides for budworm.
Right. I was just sort of thinking of an example.
Yeah. I guess I'm getting down the rabbit hole. So yeah, theoretically possible, but I don't know if it would be effective.
Thank you.
No problem.
Awesome. Greg says, I may have missed it, but I'm curious about the non-target kill of insects in the tree canopy. Is there any information about this yet?
Nothing from Nova Scotia yet. There's 2 graduate students in Nova Scotia that are looking at the beetle communities in the hemlock stands that have been treated and not treated, and another one looking at the pollinator community. So they're looking at residues within pollen and effects on the pollinator community. So that work is ongoing in Nova Scotia. I think there is some work in the US that has showed that there are some slight effects, some slight changes. So it's changes in abundance, but not community, that kind of thing.
Awesome. And Jane is saying the Zytex basil bark spray requires special permitting in NS at the moment, I think. I think it's just confirming that.
It does, yeah. So it's not actually registered for use in Canada. We're able to use it under a research permit to provide data to PMRA for its potential registration for wider use. But right now, it's you can't get it unless it's part of a research project, or you can't use it unless it's part of a research project legally.
And it's so stem injection is the only thing that landowners can use right now.
Yeah, and it's quite expensive.
Awesome. And I just had a curiosity question for myself here as well. You said that there was evidence that it took like a year for the imidacloprid to take effect. Did they state any reason why it was a year, or is that just kind of what they found there?
It's because of how long it takes the tree to move it. So it's entirely about, it's applied in the fall. The tree, it has to get into the tree, and then the tree has to move it up to the where the adelgids are feeding. And imidacloprid doesn't move very fast in the trees. There are some other insecticides that do move a lot faster. So dimethyl is one that's used in the US, but it's not registered for use in Canada, and there's no indication that it will be in the near future.
Awesome, thank you. Greg is also wondering, along with Sarah's wondering it as well, is can these chemicals be transported through mycorrhizal networks, and can it disrupt their function?
Yes, like theoretically, yes, it could be moved through the mycorrhizal networks. And there is potential for it to, there is risk that it could have an effect on the mycorrhizal community. There's some work that is being done on that in the US, and I don't think the results are conclusive yet to show what's happening, but it would be present, and it could be, it's possible for it to happen, I guess, is the right answer.
Yeah, I think actually one of our board members, Chris, Keith Egger, who's a mycologist, I know he's been doing some research on the treatment site. I think somebody, if it's not Keith, it's somebody else that they're looking at that in Kedgenkujik National Park.
Him and Donna Crossland are.
Right. Okay.
Yeah.
Is it Keith Egger that's working with him, Sarah?
From out in BC. Yeah.
Right. Yeah.
Awesome. So I'm not seeing any more questions in the chat. Chris's info is up on the screen as well, like you said before. If you have any last minute questions or want to reach out,
Can I ask one more, Morgan?
Oh, absolutely, yeah.
Yeah, I was just, so when you did your risk quotients and they all showed some risk, does that trigger more research on the back of this?
Yeah.
What does that look like?
Sorry?
What does that look like? What?
Well, so that what that means is that so if the risk quotient, it doesn't predict effects, right? So what it means is that the values are greater than 1. So that means that the work that the graduate students from Acadia are doing in the insect communities is quite important, because those are the downstream effects that we would expect to see. The other thing is that it means that we should continue to monitor and know how fast and how far away from the tree it's moving. So that's adding that 200 meter and that 400 meter piece, and how long it's persisting in the soil. Those would be the two big things. It also means that, one of the recommendations that I have for this work going forward, and for the potential treatment of further trees in Nova Scotia, is to include within that monitoring work that's going to be intensively focused on things like soil microorganisms, soil arthropods, that type of thing, where that exposure risk would be high, to be that early warning sign of, no, like we have some risk and something's happening, so we need to rethink this and have a different approach. That's, I think, what it means. You can't, the issue with these values is you can't use them to say something will happen. You can only use them to say that, yes, we should be more aware and we should keep an eye on it. Okay.
That makes sense. Thank you.
Awesome. And there's another message in the chat now. And it just says, my concern is if I treat my hemlocks, I may not know which ones are naturally resistant to this pest. Can you offer any advice on that there, Chris?
I don't think any of them are, as the sad truth. There's very little natural genetic resistance to HWA. So it's very much like a possibility, but unlikely or not found yet.
Awesome. Yeah. So yeah, I think that brings us to the end of our questions that I have on my screen here. I just want to take this opportunity to thank Chris and thank everyone for joining us tonight. This was our last talk for our Sit Back seminar series, and in July and August, we will be switching to our summer seminar series, which is a weekly series. So next Thursday, July 7th at 7 p.m., we'll be joined by Houston Grimshaw-Surrett from the Nova Scotia Invasive Species Council and Cole Vale, a student researcher at Dalhousie University and MTRI. They will be presenting on dog strangling vine in Kemptville, Nova Scotia, a threat to biodiversity and the monarch butterfly. I would like to thank the Region of Queens for supporting us and our seminar series. As always, you can stay up to date with our seminars by following us on Facebook or checking out our YouTube channel. We hope everyone stays well and that we all get to see you soon again. Thank you.